ourselves back together now. What's your vision?
As for today's video, we will be discussing about the reproduction of organisms and their cells. So, reproduction refers to the manner in which organisms reproduce, and cell reproduction refers to the process by which individual cells reproduce. So that's the definition of reproduction and the cell reproduction. There are two types of reproduction. First is we have the asexual reproduction, and second is the sexual reproduction. So we will be discussing first about the asexual reproduction. So in asexual reproduction, it produces offspring that are genetically identified to the parent, because if the offspring are all clones of the original parent. So in asexual reproduction, it composed of only one cell, which is the parent or the single parent, and a single individual can produce offspring asexually, and large numbers of offspring can be produced quickly. So in asexual reproduction, it occurs in a prokaryotic organisms, which is those bacteria, and in some eukaryotic single-celled and multi-celled organisms. As we go on to sexual reproduction, during sexual reproduction, the genetic materials of two individuals is combined to produce genetically diverse offspring that differ from their parents. When we say diverse, it is different. And the genetic diversity of sexually produced offspring is thought to give species a better chance of surviving in an unpredictable or changing environment. Sexual reproduction is the combination of usually haploid. Haploid is forming another cells, reproductive cells from two individuals to form a third, usually the diploid, unique offspring. Sexual reproduction produces offspring with novel combinations of genes. This can be adaptive advantage in unstable or unpredictable environment. Cell reproduction, it composes of eukaryotic cell reproduction and the prokaryotic cell reproduction. Eukaryotic cell reproduction is quite simple when compared with eukaryotic cell division. Prokaryotic cells reproduce by a process known as binary fission, in which one cell, which is the parent cell, splits in half to become two daughter cells. A prokaryotic cell can divide in half. Its chromosomes must be duplicated first, a process known as the DNA replication, so that each daughter cell will possess the same genetic information as the parent cell. The time it takes for binary fission to occur, example is the time it takes for one prokaryotic cell to become two cells, is called the generation time. The generation time varies from one bacterial species to another and also depends on the growth condition, which is the pH, temperature, and the availability of the nutrients. In the laboratory, which is the vitro, under the ideal condition, the E. coli, or also known as the Escherichia coli, is the type of bacteria that normally lives in your intestines. Most of these types are harmless and even help to keep digestive tract healthy. So, in a generation time of about 20 minutes, the number of cells will double every 20 minutes. Bacterial generation times range from as short as 10 minutes to as long as 24 hours or even longer in some cases. Remember that the length of time it takes for one bacteria cell to split into two cells is referred to as the organism's generation time. Is all about taxonomy. According to Berger's Manual of Systematic Bacteriology, taxonomy, the science of classification of living organisms, is the practice of identifying different organisms, classifying them into categories, and naming them. It consists of three separate but interrelated areas: the classification, nomenclature, and identification. First is we have the classification. It is the arrangement of organisms into taxonomic groups, known as taxa. In singular form, it is taxon, on the basis of similarities or relationships. Taxa includes kingdoms or domains having divisions or phyla, classes, orders, families, genera, and species. This is a closely related organisms. Organisms having a similar characteristics are placed into the same taxon. And is we have the nomenclature. It is the assignment of names to various taxa according to international rules. It is a system of giving taxonames. Example of this is calling humans as a Homo sapiens. Third is we have the identification. It is the process of determining whether an isolate belongs to one of the established named or represents a previously unidentified species. So the term to identify organisms means to learn the organism's species name. An organism's complete collection of physical characteristics is known the organism of phenotype. Remember this, an organism's that complete collection of genes is referred to as the organism's genotype, or also called the genome, and the organisms that is complete collection of physical characteristics is known as the organism's phenotype. They are different. The genes, collection of genes referred to the genotype, and collections of physical characteristics referred to as phenotype.